In Chapter 7, we're going to talk about the techniques of genetic analysis. You can think of these as the tools used in the laboratory to analyze DNA, RNAs, or proteins. So we'll be looking at various different techniques such as Western blotting, Southern blotting, PCR, hybridization, and we'll go through and talk about how these are accomplished so that if you're given a question on the test, you'll be able to adequately analyze the data you're given and come to a logical conclusion. This is how most of these questions would be presented to you. Let's begin. So we'll first look at blotting techniques. Now, the idea behind a blotting technique, if you're trying to analyze DNA, because DNA is such a large molecule, you have to chop it up into smaller pieces. And remember that we used restriction enzymes as the tools to cut the DNA into these smaller fragments. And we call these fragments of DNA restriction fragments. Now, depending upon which restriction enzyme is used, you're going to get a different pattern and different sizes of fragments after the cleavage event happens. So, for RNA and protein, these are smaller molecules. If you remember from the beginning, the DNA, if you're looking at an entire chromosome, it's 100 million base pairs, whereas RNA, it's going to be on the order of 1,000 to maybe 10,000 base pairs in length. So it's small enough to analyze directly and does not have to be cut up. Protein, by the same token, is usually small enough. Now, sometimes the protein will have to be denatured to make it easier to analyze, but it doesn't have to be proteolyzed in order to observe it on some sort of blotting technique. Now, for our purposes, we're going to start with a DNA blot. Let's see how this works. Typically, what's going to happen is the DNA will be loaded into lanes on an electrophoretic apparatus. It will be loaded into lanes within agarose or polyacrylamide gel, some sort of matrix that the nucleic acid is going to have to move through and migrate through as it moves through an electric field. Now, what charge does DNA have? It has a negative charge. So if you put DNA into an electric field, it's going to want to migrate towards the positive end of that field. And that's how electrophoresis works. Now, if you're loading the DNA into a matrix that it has to move through, you could think of the smaller molecules of DNA are just going to dart through those little matrices, and they're very quickly going to migrate from the negative end towards the positive end of the gel. If you're a larger DNA fragment, you're going to sort of bump into the matrix, and you're going to slowly move through the gel. So what that means is, when you separate nucleic acid on a gel, and you'll see this as we look, it gets separated from the largest fragment to the smallest fragment. The largest fragments are going to be on closer to the negative end of the electric field. The smaller fragments are going to zip through more quickly, and they're going to be located at the positive end of the electric field down here. So we've effectively separated our nucleic acid from largest all the way down to the smallest material. The next step then is to transfer or blot this to some sort of membrane, because we can't work with the messy agarose or polyacrylamide gel. We have to transfer this to a membrane, usually made out of nylon or nitrocellulose, some sort of solid, stable material that the DNA, through capillary action, will move over to this blot. Now, what you do in reality is you stack some the blot on top of this, some paper towels, maybe a big heavy book, like your biochemistry book. Make sure to dust it off first before you set it on there. Okay, you need some sort of weight, and in the end, through capillary action, the nucleic acid is going to move over to this blot. Now, before we can analyze any of the information over here, I want to give you an example of how we would do this. To illustrate this, I'm going to give you the beta globin gene. And I'm going to tell you that when you use a specific restriction enzyme to cut the beta globin gene, a restriction enzyme like EcoR1, I'm going to say that in the area of the beta globin gene, EcoR1 is going to cut the DNA once, twice, 3, 4 times. It's going to cut the DNA 4 times, and how many fragments will that release? 1, 2, 3. 3 fragments. Now, when you run this DNA nucleic acid out on the gel, you can't visualize these individual bands. As a matter of fact, if you stain this gel, what you would see is a smear of nucleic acid. Nucleic acid of various sizes is going to end up as a smear on that gel. Now, typically what you want to do is to visualize the gene that you are interested in studying. In this case, we're studying the beta globin gene. So what we have to do is to design a probe. This word probe comes to visit us again. What a probe is it's complementary to the gene sequence that you're interested in looking at. So you have to make the probe complementary to the gene that you want to study. So what that means is if you run all the DNA out on a gel and you denature it so that the DNA becomes single-stranded, and then you add in a complementary probe, the probe will hybridize with its target sequence. Now, you're going to want to label the probe in some way, either through radioactive means or non-radioactive fluorescent means, so that you can then later detect where that probe bound to the target DNA sequence. So, coming back to our picture, let's say that we have a probe, and we're going to make this probe specific for this entire DNA sequence that we're analyzing. So, this probe will hybridize with this fragment because it has complementary sequence. It will hybridize with this second medium-sized fragment because there's complementary sequence. And it will hybridize with the larger fragment because there's complementary sequence. Now, the next step is the hybridization. We've done the blotting. Now we're going to do the hybridization, which is when we add our probe, and that will bind to and hybridize with the bands that we're interested in. Let's visualize this. So we'll now add the probe. We've made the probe radioactive. We've made it radioactive. It's going to come in, and it's going to hybridize with target sequence. Now, it would be really nice if, as we were doing this experiment, we could actually see this happening, but you can't. You can't see this happening. You actually have to do radiography later on to visualize where the bands where the probes had bound to the appropriate sized fragments. So now we can say from this, after we do the experiment, we might do it at different temperatures so that actually at the highest temperature makes for the most strict binding. Okay, that's a point that you might be tested on. For example, if you did your hybridization at 45 degrees Celsius compared to 65 degrees Celsius, well, the binding, the hybridization that occurs at 65 degrees Celsius is way more specific than the binding that would occur at 45 degrees Celsius. We call that stringency. The 65 degrees Celsius hybridization is much more stringent than the 45 degrees Celsius hybridization. All right, now, once we do autoradiography, this would just be a blank blot on your nylon nitrocellulose, but you put some X-ray film in front of it. If you have a radioactive isotope, you will be able to visualize these bands. And now, from this information, you can relate this back to what you had with this map. You could see that, of course, the largest fragment here would correspond to this large fragment down here. The medium-sized fragment would correspond to the medium-sized fragment right here. And finally, the small fragment that migrated the most quickly through your electrophoretic field is going to correspond to this small fragment. Typically, what would be done is that standards would, a ladder, some sort of DNA ladder would be ran concomitantly with this so that you could then directly compare and say exactly what size that particular band is. That's how southern, in this case, because of DNA, southern blotting and hybridization work. It's an older technique used to map out DNA and to map out genes. Now that DNA sequencing and the Human Genome Project are so ever present, it's a little bit older technique, but still sometimes utilized and definitely tested on the USMLE. So what you need to be able to do is to, as we're looking here, is to translate the information that you see on a map down here to what you might see on a blot. You might be given a blot and asked to interpret it and be able to work backwards or forwards and say what the map would look like from that particular blot. That's the chore you might have to accomplish. So now, here's a table comparing the types of blots that you might see on the exam. Okay, we have the southern blot. This was the first one characterized. It was characterized by a scientist named E.M. Southern, so they called it the southern blot. He discovered this as a way to analyze DNA. As we'd said, you use electrophoresis to separate the bands based on size. He used a P32, an isotypically labeled probe, and this helped determine which fragments of DNA are associated with a particular gene. Which fragments are associated with the particular gene. Now, when another scientist came along and discovered a way to look at RNA, because they had a southern blot, they called it a northern blot. Right? So the northern blot is simply a way to analyze RNA, very similar in technique to the southern blot. You separate the RNA based on size. You can use a P32 labeled probe. But what a northern blot measures is the size and amounts of messenger RNA. So it doesn't tell you anything about the gene sequence per se. It actually tells you answers questions about gene expression. So it tells you whether a particular gene is being transcribed into messenger RNA. So it's a little bit more of a specific technique, actually, the northern blot than is the southern blot. Now, because scientists are so clever, when they've figured out a way to analyze proteins, they call it a western blot. So a western blot analyzes proteins. Once again, you're going to separate the proteins. You're going to separate them based on size. Now, typically what's done in order to mask the charge, the DNA has negative charges. Proteins are going to have either positive or negative charges depending on the protein. To get around that, typically in a western blot, you'll coat the protein with a very highly negatively charged molecule. So you pretty much mask any of the intrinsic charge, innate charge of the protein. You use something like sodium dodecyl sulfate. It's a very negatively charged, kind of like a soap, and it negatively coats the protein and makes it have a negative charge. This allows the protein to be separated in the electrophoretic field based on size. How do you detect? Well, you could use a radio-labeled probe, a radio-labeled antibody, but most of the time nowadays we use enzyme-linked antibodies, right? Enzyme-linked antibodies, which we can then use some colorimetric test to detect whether or not the protein was there, and we can actually use it to quantify how much protein is there. Where is this used? Well, this is used extremely commonly in the diagnostic lab. It's used to measure the amounts of antigen that might be present in a patient's blood, or it's used to measure the amount of antibody present in a patient's serum. So it can be used either way. You could use pre-made antibodies to detect protein in a patient's blood, or you could use a protein to detect the amount of antibody in a patient's serum. So this is how, for example, the ELISA test, the ELISA enzyme-linked immunosorbent assay, is sort of a spin-off of the Western blot. The ELISA is actually a little bit easier to perform. All right, now the next on here is a dot blot or a slot blot. I think of this as a quick and dirty way to look at RNA, DNA, or protein. There's no electrophoresis required. As a matter of fact, just like its name suggests, you're going to take a piece of nitrocellulose and just dot. You're going to put a little dot of DNA, RNA, or protein. Or you're just going to load it in a slot. I would think more in terms of the dot blot. That's the one you're likely to be asked about. The probe used, the same as above. You can use radioactive DNA. You could use radioactive or enzyme-linked antibodies. And it's going to detect whether the DNA, the RNA, the protein, or the antibody is there. It's just going to give you like a yes-no answer. It's not necessarily as informational as the other blot types. Why? Because you're not separating things based on charge. You're not separating the bands based on their size. So it's going to give you a quick and dirty yes or no answer. One other type of blot that I will mention because it's fair game for the examination is a hybrid of these. So we have the southern blot and we have the western blot. Now we have the southwestern blot. Okay, so what does a southwestern blot look for? It looks for DNA binding proteins. So actually for a southwestern blot, what's done is that the DNA, like in a southern blot, is separated on a gel based on size. And then proteins, DNA binding proteins, are allowed to interact with that DNA. Now, in order to detect the proteins that might interact with the DNA, what's done next is, of course, antibodies are then used to try to bind to their representative proteins. So in that way, you could detect a protein that is a DNA binding protein. You could definitely be asked about transcriptional activators, things that we talked about, leucine zipper proteins, helix turn helix proteins, zinc finger proteins, rel domain proteins. You could be asked an experiment where it's accomplished as I just mentioned, and be asked to sort of say what type of binding is going on. Well, in the case of a southwestern blot, it's a DNA binding protein. They have other types of blots. They have far eastern blots, far western blots. They even have a middle eastern blot. All sorts of different blots. You won't have to worry about those for the test, though. Another popular question is to ask you about restriction fragment length polymorphisms. So, a restriction fragment length polymorphism is pretty much like a southern blot. Okay, it's a southern blot where you're simply looking at patterns that restriction enzymes cleave the DNA into, patterns of banding. So, for RFLP analysis on a southern blot, it would be accomplished as we're showing you here. Now, this is a particular sequence of DNA, and this sequence of DNA, among all the other sequences of DNA, is going to be cleaved by a restriction enzyme. In this case, we're going to use EcoR1. Now, keep in mind, what makes one individual different from another, genetically speaking, are various polymorphisms. Now, DNA replication isn't perfect. Okay, it's really good, but it's not perfect. So as you replicate your DNA, there's going to be slight changes. And as you make your germ cells, there's going to be slight changes. So as you pass your genetic information to your children, first of all, it's going to be a mixture of yours and your spouse's or your mate's DNA that come together homologously, and they're going to recombine to make offspring. So there's going to be changes that happen from one generation to the next. The idea being this: the more genetically related an individual is to another person, the less changes they're going to have, the less polymorphisms they're going to have in their DNA. Whereas if two people are completely genetically unrelated, they're going to have quite different polymorphisms. So we can use that to our advantage to differentiate one person's DNA from another. This is the type of thing they're going to use on CSI to say whether the victim of the crime was actually, or I should say the victim, usually they know who the victim is. They're going to say whether the perpetrator, the guilty party of the crime, was actually at the scene of the crime. They're going to do that based on these RFLP analysis. Okay, so how is this accomplished? As I had said, each individual is going to have polymorphisms. Now, every person should have 2 copies of each chromosome. One they inherited from the maternal side, and one they inherited from the paternal side. Now, let's assume that we cleaved this individual's DNA with the restriction enzyme EcoR1. Now, EcoR1 is going to cleave at its palindrome at once, twice, 3 times. on this particular copy of the chromosome. So it's going to cleave 3 times. We're just using this yellow to indicate that it's being cleaved approximately at this site. Not exactly, but approximately. Now, the other copy, this individual's other copy of their chromosome, okay, is going to be cleaved differently based on mutations that we're about to talk about. Now, look at this particular A right here. This fragment here is going to correspond to the 1.2 kilobase fragment that we're going to sort of map out here. This shorter fragment is going to correspond to this 900 base or 0.9 kilobase fragment that we see up here. What happens if, because there was a polymorphism, perhaps from the paternal side, this individual inherited this particular chromosome, and from the maternal side, they inherited this particular chromosome, and there was a polymorphism or a mutation at that particular location in the DNA. Well, what does that mean for EcoR1 cleavage sites? Is this a palindrome? Well, absolutely not. This is no longer a palindrome. So when EcoR1 sees this, it's going to cleave it not into 3, but just into one big fragment. It's not going to cleave it 3 times. It's just going to cleave it twice, giving one big fragment. Now, if you add 0.9 to 1.2, it's going to be approximately 2.1 kilobases in size. Okay. Now, when we use a probe, we're going to make that probe specific to some of the DNA sequence within that particular region we're interested in studying. And depending upon how we design the probe, that's going to dictate the banding pattern we see on the southern blot. So, if we make a probe specific for this particular area of either one of these chromosomes, right? These chromosomes are homologous but very close in structure, so the probe will bind to either one. We're going to get one of the 2 following banding patterns as a result. Now, the probe, as I said, is going to determine which DNA fragments we see on the eventual southern blot. You could see that this probe is going to complementarily base pair with chromosome A up here in the following way. So you should see a 1.2 kilobase fragment somewhere in the southern blot. The probe is also going to complementarily base pair with this sequence down here. So somewhere in the southern blot, we should see a 2.1 kilobase fragment. So for this particular individual, what should their southern blot look like? Which one of these 6 lanes should it correspond to? Well, clearly it's not this choice, because this choice only has 2 of the 1.2 kilobase fragments. Actually, an individual with this, instead of having the mutation here, both of their copies of chromosome, this particular chromosome, would look like chromosome A here. It would have 2 1.2 kilobase pair fragments. This would actually be what this particular individual looks like. You see that they would have one of the 1.2 kilobase fragments, that corresponds to the ladder over here, 1.2 kilobase, and they would have one of the 2.1 kilobase fragments. So this is what this individual's banding pattern would look like. Now, what you should understand is that this probably indicates that this patient is heterozygous for a particular mutation. They're heterozygous, meaning they have one normal copy or normal allele, and then they have one mutant allele for this particular mutation. Okay? That's the transition that you need to be able to say is based on this banding pattern. You can work backwards and say, aha, they had a mutation. Now, sometimes the RFLPs are in coding sequences for important genes. Sometimes we're just looking at unimportant regions of genes. But if you're trying to do this type of analysis to look at a specific disease, you're probably going to look in the coding region. If you're trying to use this type of analysis just to look at how related one individual is to another, you'll probably want to look at non-coding sequences because you see more polymorphisms in the non-coding sequences. Getting back to our banding here. This last individual, what would they look like genetically? Well, they only have the 2.1 kilobase fragment. So this particular individual, right, is like this. They're going to have 2 copies of chromosome B. So actually, they're homozygous, and if this is a recessive type of disease, they would be homozygous recessive. They would perhaps likely have the disease. So they only have the 2.1 kilobase fragment. That brings up an important point, and that is the following. You notice how thin these bands are compared to the thickness of the bands over here. The thickness of the band basically tells you how much of that genetic information is there. So the thin bands over here compared to the thick bands mean that there's less of this 2.1 kilobase band than there is over here. And that makes sense, because in the heterozygous situation, there was only one of the 2.1 kilobase bands, whereas in the homozygous situation, there was 2 copies of the 2.1 kilobase band, thus making this thicker. Now over here, in the homozygous normal alleled person, there was 2 copies of the 1.2 kilobase fragment, which is why there is a thicker 1.2 kilobase band compared to the heterozygote over here. All right. Let's look at a different situation. Let's look at the gel that's over here on the left now. Could we use a probe that would give us that particular banding pattern? And the answer is yes. How would we have to design that probe? We're going to do the experiment the same way. We're going to cleave with EcoR1 in the same individual. It would cleave the normal allele once, twice, 3 times. In the mutant, it would cleave it once, twice. If we used a probe specific here, specific for the entire sequence, then that probe would bind to what? Remember, the probe determines which DNA fragments are seen. So this particular probe is going to bind to the 1.2 kilobase and to the 0.9 kilobase. In addition, it's also going to bind to the 2.1 kilobase fragment because it's complementary to both of these sequences on both the paternal perhaps and the maternal allele. Now, looking down here, what individual would look like this particular blot? Well, it would be an individual that is homozygous for the normal allele, chromosome A. There would be 2 copies of the 1.2 and 2 copies of the 0.9 kilobase fragment. So there would be 2 thick bands, meaning there were 2 copies. What would this particular individual, as we've shown you in this example, look like? The heterozygote. What would they look like? Well, they would be, because they're heterozygous, you would see the 1.2, the 0.9, and from their other chromosome, homologous chromosome, you would see a 2.1 kilobase band. So that corresponds to the 2.1, the 1.2, and the 0.9. That's what the heterozygote would look like. What would the homozygous recessive, in this particular case, look like? Well, they would only have the 2.1 kilobase fragment. They would not have the 1.2 or the 0.9. They would have a thick band here at 2.1 kilobases because that indicates there were 2 copies of that particular fragment. So as you can see, based on the size of the probe with respect to the gene sequence you're probing, it's going to dictate the banding patterns. So make sure you understand that where you make that probe specific to is going to be how the banding pattern appears in the final blot. Now let's look at another example of this where we're specifically looking at a disease. And in this case, we're going to look at sickle cell anemia. And I'm going to show you a way that sickle cell anemia can be screened for. Nowadays, we do hemoglobin electrophoresis and other better techniques to analyze sickle cell anemia. But this is one way you could be asked about it. It's an older way to look for sickle cell anemia. All right. So in sickle cell anemia, you know that the beta globin gene has been mutated. And actually, it's position 6 where there's a glutamate to valine substitution. And this is caused, this amino acid substitution is actually caused because there's an A that gets mutated to a T. So what type of mutation is this? This is a missense mutation, right? Because the A to T mutation now causes valine to be put in instead of glutamate. Luckily for us, this particular mutation, this A to T mutation, it actually causes the destruction of a restriction enzyme cleavage site. Now, you don't have to remember the restriction enzyme. It happens to be called MST2, but typically MST2 can cleave right at that spot where the most common mutation causing sickle cell anemia occurs. So when the A gets mutated to a T, it destroys the palindrome. So MST2 no longer can recognize that cleavage sequence because it's not there. Let's look. So in an individual that's a carrier for this particular mutation causing sickle cell anemia, they're going to have one normal allele, and they're also going to have a sickle cell allele. Now, if you look at the map for the beta globin gene, there's 1, 2, 3 exons that are interrupted by 2 introns. Okay, for both copies, the normal and the sickle cell allele, that's what you see. And that right upstream here, towards the 5' end of the coding sequence, coding strand, you're going to see that there's, of course, a mutation that happens. Because the 5' end of the coding strand is going to correspond to position 6, which is very close to the N-terminus of the encoded protein, beta globin. As I said, in the normal allele, there is a cleavage site for MST2. But in the sickle cell allele, there's a mutation here, so MST2 can no longer cleave. MST2 will cleave out here, and in the normal allele will cleave in here, but in the mutated situation can only cleave out here and over here. So, as we see with MST2, you typically get 3 cleavage events in the normal allele and 2 cleavage events in the mutant allele. If you make a probe specific for this particular sequence right here, so this probe will come in and complementarily base pair with either this sequence or this sequence down here. Okay, so it'll pair with this sequence, so you'll see a 1.15 kilobase pair band, and it'll also base pair with this sequence down here, so you, in this case, will see a 1.35 kilobase pair band. Why? Because it's had this restriction enzyme cleavage sequence removed. Because it's had this removed, you'll only see the 1.35 kilobase band with the mutant allele. Now, how does that correspond to what we're going to see in the gel later on? So if we're going to look at how the cutting works with respect to how the banding pattern is going to be, we can do the following. We can see that the normal allele down here is going to correspond to the 1.15 kilobase pair band. So this 1.15 kilobase pair band is going to correspond to the normal allele. Whereas the sickle cell allele is going to correspond to the 1.35 kilobase band. You see, the 1.35 kilobase band down here is going to correspond to this sickle cell allele. So if a particular individual's banding pattern came up like this, you need to be able to work backwards and say, aha, this person is heterozygous for the sickle cell allele. What would a normal individual look like? Well, that would be this particular individual here. This individual has a very thick 1.15 kilobase band, and that 1.15 kilobase band is, of course, indicating that they have 2 copies of the normal allele. They don't have the sickle cell allele, so they don't have a 1.35 kilobase band. So you can safely say that this individual is normal. They have 2 normal copies of the sickle cell allele. What about a diseased individual? Well, sickle cell allele is an autosomal recessive disorder, so when you look at the diseased person, you would expect that they would have 2 copies of the sickle cell allele. So they would have 2 copies of the 1.35 kilobase band. Let's look down here at that particular individual, and you see, yes, this person has a very thick, meaning there's 2 copies, of the 1.35 kilobase band. What that means is they have 2 sickle cell alleles, and actually, according to the pedigree chart here, you could see that they're probably affected. Their box is colored in. From a pedigree standpoint, that means that they have phenotypically the disease. Now, you'll notice in this particular genetic cross, you had a heterozygous father and a heterozygous mother coming together, and of course, 1 in 4 times with this particular cross, the individual is going to be diseased. 1 in 4 times, this individual is going to have a completely normal allele, 2 copies of the normal allele, and of course, 2 out of 4, or 1 in 2 times, the person is going to be heterozygous from this mating. So this particular fetus, from this banding pattern, we can see is heterozygous. You need to be able to understand pedigrees and relate to how that would work with banding patterns and, of course, the restriction enzyme maps, because that's what they like to do on the board exams. Make sure you understand how a pedigree works, and make sure you understand how southern blotting works, and that you can interrelate that information. Now, one other point, and that is, if this were an autosomal dominant disease, don't forget that in an autosomal dominant disease, 1 copy of the mutant allele is enough to cause you to phenotypically have the disease. So if this were an autosomal dominant disease like Marfan syndrome or osteogenesis imperfecta, both this parent and this parent would have the disease because they have just 1 copy of the mutant allele. In an autosomal dominant disease, assuming complete penetrance, that's enough to give you the disease. Same for this individual over here. In an autosomal dominant disease, the only normal individual would be this one right down here that has the 2 normal copies of this particular allele. Let's now look at a northern blot. A northern blot, if you remember, is a way to analyze RNA information. And what it does is it provides us information about transcription or gene expression in a particular cell type. Not only within a particular organism, but then more specifically within a particular tissue or cell type. Let's look at how this is done. Now, in this particular example, we're going to look at a northern blot that's used to analyze a specific gene called the FMR1 gene. Can you remember what that stands for? That's the familial mental retardation 1 gene. And actually, this is where you see mutations that cause Fragile X syndrome. So this is obviously carried on the X chromosome. This is a northern blot of the FMR1 gene. So what does the northern blot measure? It measures, of course, expression levels of messenger RNA, mRNA expression levels. So you see in skeletal muscle, in brain, liver, testes, lung, pancreas, and heart, each of these individual tissues was degraded, messenger RNA was harvested from those tissues, and then a probe specific for the FMR1 messenger RNA was utilized to see how much mRNA was present in each of those tissues. Now, what sorts of questions can we answer using a northern blot? I think if we go through this, this will illustrate exactly how a northern blot is utilized. Is this gene expressed in the brain? Now, based on this particular northern blot, what would your answer to that be? You go to the brain tissue, and you see a nice thick band. So do you think that the FMR1 gene is expressed in the brain? Of course the answer is yes, there's a band there. And furthermore, you shouldn't be surprised that there's expression of the FMR1 gene in the brain, because if there's a mutation to that, it causes mental retardation and macroorchidism and so forth, the other things that go along with FMR1 mutation or Fragile X syndrome. So the answer is yes. Is the gene expressed in the liver? Let's go back to the blot, and you see no signal here in the liver, no band. Is this gene expressed in the liver? Well, the answer is no. There's no gene expression in the liver. Otherwise, we would see a nice band here at the level of the liver. Why is there a dark signal for the brain compared to the lung? Now, obviously, clearly, FMR1 gene is expressed in both the brain and the lung. Why is the signal so much darker here than over here? Can you think of an answer to that? Well, it's obvious now that the brain is going to express more of this into messenger RNA and then protein than the lung. The same size band, but a thicker signal, meaning there's more gene expression of this particular gene in the brain as compared to the lung. All right, now let's look at the fact, is the gene present in the liver? Is there an FMR1 gene in the liver? You go to the liver and you don't see a signal. So what's your answer? Is this gene present in the liver? That's kind of a trick question, isn't it? Because northern blots don't tell you whether the gene's there or not. It simply tells you about expression levels. So if this is the same organism that all this tissue was derived from, then the answer is yes. If this is from the same person, then yes, the gene has to be present in the liver, because the same genes are present in all of our tissues, whether it's muscle, brain, liver, testes, lung, pancreas, heart, kidneys, whatever. The gene has to be present. If you wanted to prove it, you would have to do the southern blot, because the southern blot is what you use to detect genes. Finally, why is there a smaller size in the heart compared to the other tissues? Why is there a smaller size in the heart compared to these other tissues? That's an interesting question, isn't it? Is there a mechanism that we discussed in another chapter that could answer this for you? And actually the answer is differential RNA splicing. If you remember, alternative mRNA splicing is a phenomenon that can happen in eukaryotes. That's probably what's going on here. Okay, so let's look. Possible alternative splicing. So perhaps in the brain, the testes, and the lung, the RNA is spliced to generate a 4.4 kilobase fragment that gets translated into protein, whereas in the heart, this gets spliced in a different way. So certain exons get spliced out so that a smaller messenger RNA fragment is produced, and that this is then translated into a much smaller protein. So if you see a smaller fragment, it's not necessarily going to be like a mutation or something that happened. It's most likely going to be alternative RNA splicing that we're observing.